All right, so what I'm going to tell you about today is actually some work that was supported by the Predicate Hearing 19 Alliance. And it's really to try and understand the pathology of this particular condition that you're all very familiar with. So what is Predicate Hearing 19 girls clustering epilepsy, as I'll refer to it for this talk? Well, really the take-home message, you know the condition much better than me, but I guess the take-home message that I've got from looking at the literature, talking to you, is that it's a highly variable condition. And that's certainly a point that I want to return to as part of the talk, because it's certainly variable not only in patients, but also in the mice that we are characterising as well. And I'll come back to Joe's point about whether there are seizures or not in the mice as we get through the talk. It's also a very unusual inheritance pattern that causes this particular condition. So this is the typical X-linked inheritance pattern shown here on the left-hand side. We've got heterozygous carrier females who are generally unaffected or only mildly affected. They've got two X chromosomes. One of them carries the disease-causing mutation, and the other one doesn't. And when they pass that on to the individual shown as the black square, this is a male who's affected. He's XY. He's got the disease-causing mutation on the X chromosome and no second X chromosome that provides protection because he has a Y chromosome, and this results in the condition. So that's the typical X-linked inheritance. And as was mentioned earlier, this is a very unusual condition, Predicate Hearing 19. In fact, I'd go as far as to say that it's a unique genetic condition because, as shown on the right-hand side, we've got the black circle that indicates that females that carry the mutation on one of their X chromosomes are affected. And indeed, when they pass that on to males, they're generally unaffected or only very mildly affected, perhaps in some way. It's a little bit of a controversial area, but indeed it's the carrier females with one X chromosome that carries the mutation and one chromosome that does not. That is the disease-causing state. So a very unusual X-linked condition. So we've heard a lot about Predicate Hearing 19 protein already, and that was a great introduction from the first speaker. So you know already that it's a protein that sits in the cell, at the edge of the cell membrane. I'll talk more about that in a minute, but this is just a diagram now a little bit outdated because there are so many different types of mutations that have been identified over the years since Joseph first published the disease-causing original paper back in 2008. And really the take-home message from this slide is that this, on the left-hand side, you see basically the part of the protein that sits on the outside of the cell. And there are many, many mutations that localise within that region. So thinking about that and thinking about what Predicate Hearing 19 does, I want to give you a little bit of background about what this protein does within the neurons within the cell. So it belongs to a large group of proteins called cadherins. And in fact, Predicate Hearing 19 is just one member of the Predicate Hearing family, which is shown on the bottom right. And there are about 70 different proteins, most of which are expressed within the brain. They all have very similar structures and they probably all have very similar functions as well. So it's quite a complex family that we're dealing with here. And I guess one of the take-home messages from the talk is that we can't just look at Predicate Hearing 19 in isolation. We need to think about it as part of its role within this larger family. So on the top left, you can see what the proteins are doing within the cell. So all of them really have this same kind of structure and function, and they're very good at sticking cells together. So that's called homotypic cell-cell adhesion. And many of the mutations that I showed you on the previous slide map to those green areas, which are parts of the protein that sit between the cells, and they help two different cells, which are shown here on the left-hand side, to stick together. And we've known that for many years. So we did some experiments in the laboratory just to really confirm what had already been published a number of years ago. And this is that Predicate Hearing 19 mediates homotypic cell-cell adhesion. So what do I mean by that? What I mean is that it's very good at sticking cells together that express Predicate Hearing 19. So we can do these experiments within the lab by forcing cells to express predicate hearings, and I'll show these kinds of images a few times within the talk. We've got cells that express Predicate Hearing 19 in purple, in this particular on the top left, and other cells that express Predicate Hearing 19 labelled in green. So two different populations, they're essentially identical. When we put those two populations together, it's called a cell sorting assay, and we're asking the question, do they form mixed aggregates or not. And indeed they do, as you can see. Speckly patterns indicating that the aggregates contain both populations of cells. In the middle panel, we've got exactly the same experiment with another family member, protocadherin 17, and on the right-hand side, protocadherin 10. They all function the same kind of way. But what's more interesting is when you do a cell mixing experiment. So here we've mixed together protocadherin 19 expressing cells in purple and protocadherin 17 expressing cells in green on the left-hand side at the bottom, and they formed aggregates, but they're distinct from each other. They form separate aggregates. They sort out within the culture system. And that indicates that a 19 expressing cell doesn't really like to mix with a 17 expressing cell, but rather they'll just prefer to stick to the same type of cell and form separate aggregates. Same goes to 19 and 10 and 17 and 10. So that leads us to really the theory that was again alluded to earlier. How is it that this condition manifests? What is it? What's going on within the developing brain to cause the neurological symptoms associated with protocadherin epilepsy? And essentially this is the sort of long-standing theory that has been proposed a number of years ago, and the idea is as follows. On the left-hand side we have a normal individual in which there are no mutations of protocadherin 19. They are unsymptomatic, of course, and that's because the cells can form their normal connections within the developing brain. On the right-hand side, we've got shown there as mutated males in mosaic, mutated females in mosaic, mutated males. This is, if you like, the disease condition, two populations of cells, and these two different populations of cells occur because of X inactivation, so that the cells shown in the grey have the mutation in protocadherin 19, and those that are shown in white don't have the mutation. So this random X inactivation process occurs during early embryonic development, creates the two different types of cells, and because protocadherin 19 cells like to associate with other protocadherin 19 cells, it was proposed that there are two separate networks that develop during development. And this causes a scrambling of the normal neuronal connections, and you get two different populations, resulting in the epilepsy and other symptoms. And then finally, in the middle, we've got the situation of mutated males, where all of the cells are mutant, because there's only one type of population of cells. They all carry the mutation, and therefore it's restored to the normal type of neuronal activities. So that was the theory that stood for a number of years. It raises a number of questions, though, and that is, how does this mosaic expression of protocadherin 19 lead to epilepsy, and at the cellular and molecular level? So what's going on in female brains to cause this, to cause the epilepsy? And also, a more intriguing question, why does the complete loss of protocadherin 19 in males not cause epilepsy? So how can the brains develop normally in males when they lack the protein completely? And we wanted to do some experiments to try and get to the bottom of this. And without getting too technical and sort of giving you too much detail that you don't need, this is really borrowed from some papers that came out a number of, quite recently actually, and they looked at not the protocadherin subclass of protocadherins, but another large group of protocadherins that essentially have the same structure. And the bottom line here is that they didn't look at just single protocadherin molecules. They looked at the combinatorial effect of expressing many protocadherins within the same cell. And really the take-home message is that these proteins work in combination, and they form adhesion codes which are dependent on the combination of protocadherins that are expressed in individual cells. So just to give you one example, on the top left-hand side, we've now got a cell that expresses three different protocadherins called alpha, beta and gamma. The numbers aren't important. The key point here is, though, that the combination of protocadherins expressed in the red cells and the green cells are the same. And under those conditions, you get nice mixing. But as soon as you alter just one of the members expressed by the cell, you get this segregation effect, as shown here on the rest of the panels there at the top. And they did a lot of experiments and looked at this in a lot of detail. And really what the message is that you can take home from this slide is that in combination, when you consider all of the different protocadherins that are expressed in the cell, they form a lattice work that has certain rules associated with it. And this enables the cells that express exactly the same combination to stick together and form associations, whereas even a single mismatch in that code results in an inability for the cells to stick together. And this was all done in clustered protocoherons, but had not yet been done in the non-clustered protocoherons, which is the group to which protocoheron 19 belongs. So we borrowed from these papers that came out with clustered protocoherons and asked the same question of the non-clustered protocoherons and tried to understand that in the context of protocoheron 19 epilepsy. So our key questions were, do non-clustered protocoherons have overlapping expression, which is required for this combinatorial code to occur? Do they form specific trans interactions, in other words, between the cells? And do they form promiscuous cis interactions? So in other words, does the code work the same way? So the first question had really already been answered to a large degree. Many papers have looked at the expression of non-clustered protocoheron 19s, and many are shown there on the left-hand side. 3 that I'm going to focus on today are protocoheron 19, obviously at the bottom. Protocoheron 19, 10 and 17 are also in the red boxes. Basically, the message here is that they all have complex expression during the brain. So these are mouse brain sections, and you can see that stripe of expression along the spinal cord, and up at the top in each section is the developing brain. So they're all expressed within the brain, pretty much, with complex expression patterns. The trans expression is also true. So there are specific trans interactions that occur that I've shown you that already by the cell mixing experiment shown here on the right-hand side. And some biochemistry experiments that we did also showed that they have promiscuous cis interactions. So really here what I'm trying to tell you is that the code works the same way for these non-clustered protocoheron 19s as it does for the clustered protocoheron 19s, as far as we could tell from these experiments. Okay, so then could we start to use this information to understand protocoheron 19 epilepsy? So this is now an experiment, I think the first one that had been attempted, in which we've tried to put in not just protocoheron 19 into the system, but looked at it in the context of the other non-clustered protocoheron proteins. So in this experiment, we've got protocoheron 19 and 10 in the purple cells and 19 and 10 in the green cells. So we're starting to build up a combination, and the cells mix together beautifully. But if we start to play with those combinations now, and for example have purple cells that are protocoheron 19-10 positive, but green cells that are 19 and 10, so a mismatch in the two codes, they segregate away from each other. They still stick together, and sometimes they even stick together in the same aggregate, but you can see there's a nice segregation between the cells by and large. We can make it slightly different, put 19 and 17 together and 19 and 10 together, and again we see this segregation phenotype. And we can make it more complex again and put 3 different protocoheron 19s, 19, 17 and 10 in purple, 19 and 10 in the green. And again, we get this segregation phenotype. So we're looking at the difference between anything on the bottom is a mismatch, and you can see they segregate, whereas on the top you can see a perfect match and they integrate nicely. So this idea of a combinatorial code seems to stack up for the protocoheron 19-like protocoherons, which are the non-clustered protocoherons. So we wanted to take this a step further and understand it in the context of disease-causing mutations in protocoheron 19 girls clustering epilepsy. This is just a little bit of data to show you that when we look at these proteins that cause the disease using this assay, they don't have any aggregation activity at all. So the control is the top right, which says 19, and you can see the cells are clustering, the green cells. But 2 examples of disease-causing mutations are on the bottom, and you can see these like to stay as single cells. They have lost their ability to interact with each other. So they seem to be proteins that are not functional within this assay. And that's just some data to show that they're expressed. And then we quantitated the aggregation data. So now we've started to build up a model, just within cell culture, of what we could consider to be, let's test the combinatorial model in the context of cell culture studies for protocoheron 19. So here's a mimic of a normal individual. There were no protocoheron 19 mutations in this first example. Two lots of cells, both expressing 19 and 10, and you can see they mix nicely, so that's as you would expect. Here's an example of a hemizygous male modelled in culture, and here again we have cells that are labelled in red and in green, and they both express the same thing. They've got a normal protocoheron 10, and in combination with a mutated form of protocoheron 19, and again they form nice mixed aggregates as you'd expect. And this is the mimic of the disease-causing state, where we've got 2 different populations. In this case we've got 19 and 10 in green, and in combination with a protocoherin 19 expressing, a mutated cell expressing the disease-causing mutation in combination with 10 in purple. Take-home message here is that these form separate aggregates, or they sort themselves out from each other in cell culture, a different phenotype. So this does provide support for this cellular interference model. And really that's was the first time that people had sort of thought about protocoherin 19 in the context of combinatorial cell-cell adhesion. And we wanted to take this a step further. Cell culture models are great because they're easy to use, they're accessible, and you can manipulate them easily, but they're not very physiological. So we wanted to look in the context of a developing brain, what protocoherin 19 was doing, and consider it in combination with other protocoherins. And to that, we really needed to use the mouse model for that approach. So this is just a little bit of technical detail showing you that we solved a problem which was alluded to earlier, that it's very difficult, in fact impossible with the reagents we have at the moment, to detect protocoherin 19 protein that's expressed by the brain endogenously using existing antibodies. And it's one that's plagued us for many years, so we used a new technology called CRISPR to cut and insert a little tag on the endogenous protocoherin 19 gene, and that enabled us to look at the protein in vivo in its normal state. This just shows you that the approach works. So on the left-hand side of the two images, they're brain sections. This is the epitope that we inserted. It's called HA. Not so important, but really the take-home message here is that we've got no signal on the left-hand side brain, and we've got some signal on the right-hand side. And that's showing us for the first time where the protein is expressed endogenously during brain development. And you can see actually that protocoherin 19 expressing cells are quite abundant within the developing brain, and that was a really nice demonstration earlier of some of the detail associated with its expression. And essentially what we find is that the protein reflects the data that Isabella showed earlier. It resolves to layer 5A as the brain develops. Before the cortex becomes fully developed, though, there's a stage where we have an abundance of neural stem cells or progenitor cells from which the neurons are derived. And what we also find is that protocoherin 19 is expressed by those cells during development, and then it resolves to some of the excitatory neurons, and we're still resolving exactly which ones as well, and some of the inhibitory neuronal precursors as well. Again, there's a little bit of data. This is very similar to what was shown earlier, but rather than using an RNA probe to find the protocoherin cells, we're now looking at protein, but essentially the take-home message is the same. It resolves to layer 5A, and it's quite a complex heterogeneous population within that layer. Okay, so can we now use this model that we've created to understand much better what's going on in terms of the combinatorial role of protocoherin 19? So we used it to see whether we could look endogenously within the developing brain, whether there was interaction between protocoherin 19 and 10 and protocoherin 19 and 17. And we do find some evidence from that. These are technically quite challenging experiments. Anytime that you want to look at the interaction of two proteins that are expressed at their normal endogenous levels, it can be quite a difficult experiment. Nevertheless, we're reasonably well satisfied that there is interaction between protocoherin 19 and other family members within the developing brain. So this is providing support for this combinatorial model that that we proposed earlier. Now, this model has also been presented recently by Isabella. She talked about looking at fine detail at the developing cortex and looking carefully to see whether there's a normal number of cells within the heterozygous females. We published this a couple of years ago, and as she indicated, we couldn't see anything at the gross level. We didn't do the fine detail analysis that she's doing, which is great to see. But but looking at sections and by comparing the cross sections, there was nothing really obvious in terms of gross morphological features in this protocoherin 19 knockout mouse. Whether we looked at knockout males, whether we looked at heterozygous females, anything we looked at a very gross level, there was nothing obvious that we could see in terms of the brain morphology as being different. Nevertheless, it's a very useful model because it provides a way of detecting which cells express protocoherin 19 but lack a functional protein, and that's because there's a little tag in it called LAC-Z, which I won't go into the details of, but it provides a nice way of labelling the mutant cells within the developing brain. And you can see that with the blue signal shown in the right-hand side in those brain sections. So it became a very useful model for the series of experiments which I'm about to tell you. Okay, so this was kind of what we've been building towards for the last couple of years, or even longer. We wanted a system where we could detect the wild-type cells and the mutant cells within the developing brain at their endogenous level, so in other words, the protein that's produced, and we wanted to be able to track the location of the cells and ask the question, are heterozygous female brains developing differently to hemizygous male brains, or indeed wild-type brains as well? And this system that we've put together enabled us to do that experiment. So here are the results, and on the left-hand side I'm just going to show you a control to start with. So this is a brain section around 2/3 the way through mouse brain development. It's 14 and a half days, and at this stage the cortex has started to differentiate into some of the neurons that are present, and you can see those green signals there. They're the protein-adherent 19 positive cells. So this is a wild-type brain in which half the cells contain the tag, and the other half are just wild-type cells that won't be labelled. And that's indicated with that little schematic on the left-hand side. This is it's a wild-type brain, essentially, with half the cells labelled with protein-adherent 19 tag. And you can see there's lots of expression of protein-adherent 19. Now, if we compare that to the one on the right-hand side, this is a mimic of protein-adherent 19 epilepsy, where we've got a situation where we've got the wild-type cells shown in green, and the mutant cells, so those cells that have activated the chromosome which has got the mutation on it, are shown in this case as purple. You'll see the stain in a minute, but you can see the location of the wild type cells is quite different to the control on the left hand side. They've segregated away from their neighbours, and they've formed little clusters in the developing brain. Quite a profound and pronounced phenotype. This is the stain for the mutant cells in the protein-adherent 19 girls clustering epilepsy mimic. And when you put those two together, what you get is this picture of the developing cortex where we've got both lots of cells labelled, and you can see there's this segregation. There's patches of wild-type cells, patches of mutant cells, patches of wild-type cells, and this is like a zebra stripe across the developing cortex. And the control, as I said, is on the top left-hand side where we don't see this effect. This is some quantitation to show you that we see it. It's a statistically significant effect. It's mainly pronounced in the cortex, actually. That's the main site where we see this segregation during brain development. Some more examples, and this is what I want to focus in on for a moment, is to tell you about when this phenotype first occurs. So when we look at the protein expression during development, the first time we see protein-adherent 19 protein development is very early in brain development, really just about almost as soon as the brain cells have decided to become brain cells and separated away from the other ectoderm cells. And that's 9.5 days in development, which is about halfway through mouse development. So we knew that the protein-adherent 19 expression occurred very early during brain development, so we wanted to understand that in the context of this segregation phenotype that we saw. And here's a little bit of data on that. So on the left-hand side, we see the time point where protein-adherent 19 is first expressed. The mimic for girls clustering epilepsy is on the bottom left-hand side. And as the protein is expressed, we can't really see this segregation phenotype at that very early stage. But even just 1 day later, at 10.5, which is the right-hand side panels, you can see this situation here. The control is on the top, where we see sort of uniform expression of protein-adherent 19 HA signal, even though only half the cells will be expressing it on average. On the bottom right-hand side, you can see already the cells have started to separate from their neighbours. They've become separated into positive and negative clusters at this very early stage of brain development, really before even neurons have been generated. And this is just to tell you that this phenotype, this segregation that occurs, continues all the way through brain development, all the way through to postnatal. So this is postnatal day 7, so a week after birth. Again, we see this segregation. You can see the obvious difference on the bottom versus the top. And each brain is different. So this is coming back to this idea that it's such a variable phenotype in the spectrum of protein-adherent 19 girls clustering epilepsy affected girls. We know it's variable. And we also know that at the level of the mice that we look at, each brain is unique, each brain is different, and we believe this is due to the exon activation pattern, which is going to be unique to each brain. If you cast your eye across the middle set of three here, these are all at the same stage of development. I don't know if I can get this one to work, probably because it's not plugged in. There we go. So with these three across here, these are three different brains at 14 and a half days of development, all have the same genotype, so they've all got the same mixture of positive and negative cells within them, but the pattern that they've segregated into is unique for each of the brains. So we think this might be contributing to the variability that you see across the patients with this condition. Okay, so what does all this mean? We've tried to sort of formulate this into a model to give us something that we can grab onto to understand how this condition is being caused at the cellular level, and this is what we've come up with. So again, not to get into too much of the technical detail, this really reflects a situation where we've got two cells that are identical, coming together during development. The different coloured blocks are the different forms of protocadherins that are expressed, in this case clustered protocadherins, which this was based on. And you can see they form this nice lattice work, and that helps the cells to associate or stick together during development. As soon as you mess with that code, and for in this case you've got a different member indicated by the different colours, it causes the lattice work to break down. The cells don't stick together very well. And this is the situation for protocadherin epilepsy here at the bottom, where we've got one cell that expresses, for example, 3 protocadherins, and this one's missing the protocadherin 19, which would be the green one. You can see again this lattice work breaks down, and in fact it's quite a significant break if you look at it in terms of the numbers that are generated by these models. So we think there's a good basis for this kind of mechanism occurring during development. This gets quite technically challenging here, and I won't go again through into a lot of the detail, but the take-home message is that if you alter this combination of protocadherins that are expressed by 2 different cells, even by just one member, so just make a single difference between the 2, it doesn't really matter how many members of the family each of the neurons or each of the cells are expressing. As soon as you change it by 1, you get a massive drop in the size of that lattice work that can be generated, and therefore the cells would be predicted not to stick together. And that's basically what this graph is showing you. It's an in silico, so computer modelling approach. And we thought that was interesting because it gave us an idea, could we rescue, in other words, get rid of the segregation phenotype in the heterozygous females by restoring all the cells back to having the same code again. So we did that experiment again as well to try and sort of get further evidence for this model. And it was a technically quite challenging experiment, but again, not to give you, to get caught up too much in the detail of it. Essentially what we wanted to do, as I said, is to start off with cells that are heterozygous for protocadherin 19, so that's the disease-causing condition in both cases. But what we did is we added, again used CRISPR to delete the wild-type chromosome so that we made it into a mutant form of protocadherin 19. And that would mean that we've got now uniformity in the genotype. We've removed the fact that they've got 2 different types of cells being produced, and now they'll only produce mutant cells. And under those conditions, what should happen is that we get rid of that segregation phenotype because the cells now have a uniform genotype for protocadherin 19. So in other words, what we're looking for is a rescue of this phenotype to say, okay, we've got segregation here in these heterozygous brain. If we get rid of the mutant cells, if we get rid of the wild-type cells and turn them into mutant cells, we should restore that back to a normal, unsegregated brain. And indeed, that's what we saw when we did this experiment. So this was further evidence of this combinatorial code for protocadherin 19 in combination with other members of the same family. So that's all well and good. So I've shown you a lot of data looking at mouse brains, looking at sections, looking at where the proteins are and the cells are and the effect of having a heterozygous brain. But what's going on in the brains, and does that cause a phenotype? So this is coming back to Joe's question from before. Do the mice have seizures? So it's one we get asked quite often, especially by reviewers of papers that we submit. And it's not an easy one to answer in the sense that we've done some experiments in this area. They don't have frank seizures. So we can't look at these mice, monitor them over time and see consistent evidence of seizures. We don't see that. But what we have done in collaboration with a group in Melbourne, Kay Richards and Steve Petru, is that they look very carefully at the brain activity in these mice and ask the question, is there evidence of elevated brain activity, especially in heterozygous female mice that are the mimic of predicate hearing 19 epilepsy in girls? So first of all, here's a normal brain. It's a brain from a wild-type individual, so no mutation in predicate hearing 19, and this is, if you like, baseline brain activity, and that was monitored as shown on the upper right there with EKG assessment. And this is what happens in female mice that have heterozygosity, so for predicate hearing 19. So this is the mimic of predicate hearing 19 girls clustering epilepsy, and I think you can see there's quite a profound difference between the two. And what was really interesting to us was when we looked at mice that had no predicate hearing 19 present, it went back to wild type again. So we've gone from normal brain on the top, disease mimic in the middle, and then at the bottom we've gone back to what's equivalent to hemizygous males, the carrier males that aren't affected. It's actually female in this case because we wanted to keep the gender consistent. The effect, though, is the same. We find it's back to baseline level again. So really interesting EKG data indicating there's a lot going on in these brains that we can't see as frank seizures. Perhaps it's manifest in the behavioural data that was shown earlier to some degree as well. But clearly there's differences in the patterning, in the activity of the brain in the heterozygous mice. And we did some statistics to look at that. Again, look at the variability in the heterozygous females. So this is all the same genotype, but you can see there's quite a range of effectiveness in terms of this spike wave seizure discharge and the frequency of it in those individuals. Again, showing that there's variability even in mice. And then the very last point I'm going to show you is just a study that we did with Simone Mandelstam in Melbourne. She's an excellent paediatric radiologist, and this is also in combination with Joseph, who helped to coordinate this and had been looking at this for a number of years. She's been looking very carefully at the patients' MRI scans, and as MRI technology has improved, it's now possible to look more carefully and more critically at the brain development to see if there are any slight abnormalities that wouldn't have been obvious a number of years ago when the scanning wasn't as high resolution. And she's been able to look at these brains and come up with a number of relatively subtle malformations, cortical folding, slight cortical folding differences, unusual areas of sulcation, so that's the way the brain folds, focal cortical dysplasias and so on. I'm no expert in MRI, but what she certainly is, and she's indicating to us that it's variable, and occasionally she sees these unusual brain features. We don't know if they're causative or not, but I think it's worth reporting them to this group because there's probably further evidence of this coming out in the literature as well, and it might be associated with this segregation phenotype that we see in the developing mouse brains. So it's certainly an area that we're keeping an eye on. All right, so that's really all I wanted to tell you today, and I want to summarise it in this slide here. And this really describes what we've been working on for the past, 3 or 4 years, really. And we've come up really with this model now, which is shown here on the left, where in a heterozygous female brain, there are 3 different things that are going on. There are cells that have predicate hearing 19 that like to stick to each other. There are cells that don't have predicate hearing 19 that like to stick to each other. And there are cells that, at the interface of those 2 different populations, that don't like to stick to each other. And we get this segregation phenotype because of that. And as a result, when the cortex becomes fully developed, we get these patches of positive and negative cells across the developing cortex. So what the big gap in our knowledge is now is what connects this phenotype, how is it that this segregation phenotype causes that elevated brain activity that I showed you does occur in mice as well. So what is it about the segregation that's causing this apparent scrambling of the neuronal circuitry to give rise to elevated brain activity? That's really where we're trying to head in the future. That's a very difficult one to solve, but part of that is identifying exactly which circuitry predicate hearing 19 is involved with. But at least we now have some kind of cellular mechanism for this unusual and unique inheritance of predicate hearing 19 epilepsy. So I think we've made some progress along the way. It's still a very long road to get to the end of it, but we've started that journey, and I think the mouse models are giving us some real insights, and we'll continue to use them as we head along the road. So as part of the acknowledgements for this, I wanted to really point to this paper because it really was a team effort that got us to this point. We published it all, most of what I told you earlier this year, and if you were interested in looking at this paper but don't want to get through all the technical detail, you can Google PCDH19 and Node, and we wrote the sort of more blog style summary of the paper for one of the other journals that was associated with this one. It's a little bit more accessible, if you like, and you might find that an interesting thing to read. I'd also like to acknowledge, of course, all of my collaborators, Joseph for many, many years on this, and other members of the team in Melbourne particularly. And the guy on the left-hand side that you can see there, that's Daniel Pederick. He was a former PhD student. He's got his PCDH19 Alliance top on that I think I got a couple of years ago. And he did basically all of the mouse modelling work that I've been showing you. So thanks to support from the PCDH19 Alliance and other supporters as well. And with that, I'll close, and I'm happy to take any questions. Thank you.
Any questions for Paul? I have one question, actually. If you could put up 2 slides back with your summary slide. Is there any hypothesis as to the width of the segregation? Like, it does seem very abrupt, where it stops and then the new population starts. And what determines that?
The interesting thing about early cortex development is that the cells are very closely associated as they divide and then ultimately differentiate. There's a very intimate association because the scaffolding of the cells is all the way across the entire cortex. Okay, so the nuclei come up and down, there's a division step and they separate them. So there's a lot of contact between the cells. And I think because of that, there's ample opportunity for them to sample their local environment. So they're continually looking to see if the cell next door is a PCDH19 positive cell or a negative cell. So what we find is that there's scope for them to form the clusters because of the active division that's occurring during cell migration, during nuclear migration, during cortical development. Sometimes we find that it's not a perfectly radial patch and that cells will seem to be positive here and then there'll be associations across it. So we look at this in 2D sections mostly. It'd be very interesting to look at it in 3D sections to see actually the shape of some of those patches during development. But each brain is unique. Each one is different.
You showed the slide where you were seeing the difference in electrical activity. I know that seizures have not been caught, but is there any other pattern that you're able to notice in terms of cell-to-cell connectivity or spectral patterns or different frequencies between the wild types and the heterozygotes?
Come get my exercise.
Yeah, we mainly focused on spike wave discharge frequency and spike wave discharge intensity, as we thought that was the best surrogate for epilepsy. There's a lot of data there though, and we're not finished looking at it yet. These are quite long captures of activity, and we have many, many mice that were done as part of that experiment. But I would also say that it's a very difficult analysis to do because each of the females is unique. And it's not like looking at a disorder where you've got wild type knockout, 3 of each, done and dusted. The knockouts are going to be all the same in theory, and the wild types is the control. Here we've got variability in each of those females. So what you see in one female may not be present in the next one. Not saying it's not important, but there's you're looking for a consistency in features, but it's not going to be present in all of the mice. So that makes it a difficult analysis to do.
One more question.
Hi, Paul. Hi. This is a wonderful study, and I I mean, there are, of course, many things that can be asked, but I have a strong bias towards physiology. So this is what I'm really interested in. Here you show vertical stripes, and that, of course, has to do with many things, including the fact that principal neurons migrate vertically during neurogenesis and during the formation of the layering, which is certainly a process is going to be extremely important here. On the other hand, interneurons, inhibitory neurons, are migrating tangentially in the cortex. So I mean, going back to the epileptic phenotype, which of course is so important, at a certain point will become important not only to talk about the clustering of neurons, but maybe about the clustering of synapses. So what happens if an interneurons end up in a purple column or in a green column? And I mean, I think this is an incredibly interesting question, and I hope that you will really get into that at a certain point of your life.
We are trying to pursue that very question of whether interneurons are deployed in an appropriate way during cortical development. As I said, we see some predicate heron 19 expression in the interneuron precursors as they migrate tangentially. And we wonder, we don't really have evidence for this, but we wonder whether it might be that because the interneuron precursors express predicate heron 19 and at least some of ultimately the excitatory neurons express predicate heron 19, whether during homing of the interneuron precursors, they may like to associate with their like column in the radial cortex. And if that were the case, there might be a mismatch that occurs during segregation of the interneuron precursors, shown here on the top, as they migrate around. They may find themselves not aligned with the like column in the cortex. So if there was a role of predicate heron 19 in the homing mechanism, which we don't know there really is, but it's sort of an attractive hypothesis to test whether we see, therefore, misdeployment of the interneurons as they try and migrate down into their appropriate layer in the excitatory cortex, so into the cortex. So early days, one way to test this, of course, is to make a mutant patch of cortex using in utero electroporation and ask whether the interneurons that are wild type don't migrate to that mutant patch of cortex. I'm really happy to see this slide. Well, yeah, we're doing our best. Yeah.
All right, one.
Hi, I just had a question around sort of development and if you see this, changing through the development of the brain. So it looked, I don't know, one of the slides looked, and I could have misinterpreted it, that the zebra striping seems to be less as the brain gets older.
Each brain is different. And of course, we can't look at one brain as it changes across time. We just can't do that at the moment. It would be lovely to be able to do it, image them live and look at the change of the brain and how those clones develop during cortical development. But so the best we can do is look at different brains at different time points and ask the question, is the segregation that's apparent early, does it continue later on during development? And the best data I've got for that is this one, where we've got a normal odds control. So this is half the cells labelled at the top, but the interesting one is the bottom one, where we've got the patches are still there after birth. And we actually have a bit of trouble looking at 6 weeks because predicate heron expression seems to decrease a lot. But even by 6 weeks, we see that those patches are there. So in my view, what happens during cortical development persists after birth in mice.